This presentation about the pharmacodynamics is prepared by Professor Mohammed Abdelbari, Professor of Pharmacology, and Shamas Faculty of Medicine. The prepared narration is read by the Microsoft David voice. The term pharmacodynamics may be defined as the effect or the action of the drug on the body. We are interested in two main items when we discuss the pharmacodynamics of a drug. 1. The mechanism, or the mechanisms, by which the drug induces its actions. 2. What are the factors which may modify the drug actions? This will be postponed to be discussed with the pharmacotherapeutics. Let us start with the first item, the mechanisms of drug action. There are two main mechanisms by which the drug can affect the body functions. 1. The receptor mechanism of drug action, where the drug interacts with a part of the target cell, called the receptor, in order to change the function of this cell, or in other words, to induce a pharmacological action. These receptors are dedicated for binding biological molecules, we call them the ligands. The receptors do not serve any other function in the cell. We are going to discuss two processes that occur successively when the drug reaches the receptors. A. Binding of the drug to the receptors. This is the cell membrane. This is the receptor, which is usually a part of the cell membrane, although it may be inside the cell. This is the drug. The drug binds the receptor through a chemical or physical bond. The process of binding may be termed the reception. And B. The changes, which occur in the cell after binding the drug, these changes are called post-receptor signaling. This is the post-receptor target. Binding of the drug triggers a series of changes in the target. These changes end in target activation. The process of signaling may be termed the transduction. Finally, the activated target triggers activation of an effector to initiate the response. 2. The non-receptor mechanism of drug action, where the drug may either A. Bind to a target on the cell, other than the receptors. This target is not dedicated for drug binding, but is actually serving another function. Binding of the drug will modify the actual function of this target. B. The drug may not bind to a cell, but interacts, either chemically or physically, with another molecule in the medium around the cells. By the end of this lecture, you should be able to 1. Enumerate the different components of the receptor mechanisms of drug action. 2. Enumerate the type of bonds between the drug and the receptors, with examples. 3. Define the drug types with examples. 4. Draw the dose-response curves for each drug type, and deduce the possible parameters from each curve. Now, let us start with discussing the details of the receptor mechanism of drug action, under the two main categories, the drug binding, or reception, and the response signaling, or transduction. Under the category of the drug binding, we are going to discuss 4 items. First, the types of bonds between the drug, or the ligand, and the receptor. For each type, you should know the effect of this type of bond on the drug action, and at least one example. Second, the types of drugs, or the ligand. Whether it is an agonist or antagonist, and the subtype of each one. You should be able to compare between each subtype, and give at least one example for each subtype. Third, the dose-response relationship curves, for each type or subtype of the drugs, and the pharmacological significance of this relationship. You should be able to get the important parameters that can be inferred from each curve. Fourth, the theories that explain the dynamics of the drug actions. Finally, we will discuss the 5 important type of signaling mechanisms, with examples for each mechanism. Let us start with the type of bonds that links the drug to the receptors. The first type is the covalent bond. It is a stable and strong bond that needs high energy to be broken. Most commonly, it occurs between 2 carbons, which share electrons to complete their outer shells. However, it can occur with many other types of atoms, e.g. phosphorus and oxygen, 2 sulfur atoms, etc. Since the covalent bond is a very strong bond, the binding between the drug and the receptor is irreversible. Recovery can occur only by new receptors synthesis. Accordingly, the duration of action of the drug is long, and it has no relation to the drug elimination, but to the rate of receptors turnover. The most famous example for the covalent bond is between the alpha receptors and the alpha blocker, phenoxybenzamine. These type of bonding may be also utilized in drug binding to non-receptor targets, e.g. the organophosphorus compounds and the choline esterase enzyme. That is why the organophosphorus compounds are termed the irreversible anticholine esterases. Covalent bonding can also occur between the sulfur atom of the cysteine amino acid of the proton pump and the sulfur atom of sulfenamide, or the activated proton pump inhibitor. This is the base for the long duration of action of the proton pump inhibitors, despite their very short half-lives. The second type of bonds is the electrostatic, or ionic bond. It is the most common type of bonds between the drug and the receptors. It less strong than the covalent bond. The electrostatic bond occurs between 2 oppositely charged ions, e.g. a positive ion on the surface of the drug molecule, and a negative ion on the receptor. Since the energy of the ionic bond is not high as the covalent bond, it can be easily broken, and the action of the drug is reversible. The duration of the drug action depends on the drug elimination kinetics, so the duration of action is variable from one drug to another. The most famous example is the ionic bond between the positive quaternary nitrogen in the cationic head of choline esters, as acetylcholine, and negative charge on the anionic side of the muscarinic receptors. The third type of bonds between the drug and its receptors is the hydrogen bond. It is a type of the electrostatic bonds, but is much weaker. The electrostatic attraction occurs between the positively charged hydrogen ion and the negatively charged oxygen. Although the hydrogen bond is a very weak bond, yet it may be multiple, due to the presence of many hydrogen ions on the surface of the organic compounds, which makes this bond important. The most famous example is the hydrogen bond between the positive hydrogen atom in the esteritic head of choline esters, as acetylcholine, and negative charge on the oxygen atom, of the esteritic site of the muscarinic receptors. The fourth type of bonds is the van der Waals bond. It is still a type of the electrostatic bonds. And it is a very weak bond, even weaker than the hydrogen bond. Van der Waals bond occurs between very large molecules, where there is a chance for induced dipole formation. It is usually formed together with multiple hydrogen bonds, which makes the drug binding more stable. The most famous example is the bond between the anti-cancer drug imatinib and fusion gene that codes for a of tyrosine kinase in the malignant cells. This gene is called BCR-ABL gene, since it is a fusion between the breakpoint cluster region, BCR, on chromosome 22, and ABL gene, on chromosome 9. The last type of bonds is the hydrophobic bond. It occurs between two hydrophobic regions on the drug and the receptor, leaving the remaining hydrophilic part of the drug receptor complex exposed to the outside. The hydrophilic water molecules coat this complex, thus adding to the bond stability. The most famous example is between the general anesthetics and the neuronal receptors that can modify ion channels function. Thus, the action of the general anesthetics depends on the physical property of the drug, not on its chemical structure. Second, the types of the ligand, or the drug. There are two main types, the agonist and the antagonist. Sometimes we call it the blocker. Let us start with the agonist. There are three subtypes of the agonist: 1. The full agonist, 2. The partial agonist, and 3. The inverse agonist. When we use the term agonist, by default, it is the full agonist. Otherwise, when we want to describe the other two types, we have to use the full name explicitly, that is, we say the partial agonist, or the inverse agonist. The full agonist is characterized by two features, the affinity and the efficacy. If this is the receptor, and this is the agonist, the three-dimensional shape of the agonist fits well in the binding site of the receptor, like the key and the lock. This is called affinity. The binding of the agonist triggers a post-receptor signaling, which initiates the pharmacological response, or the action. This is called efficacy. There are many examples of full agonists in pharmacology, e.g. epinephrine, and the alpha or beta receptors, or the morphine, and the mu, or delta receptors. The effect, or the response of the agonist, may be 1. Quantitative effect, that is, it has a quantity, which can be measured, e.g. the degree of reduction of the blood pressure in a hypertensive patient, or the reduction in the blood glucose in a diabetic patient. 2. Qualitative effect, which can be described, but cannot be measured, e.g. termination of arrhythmia in cardiac patient, or induction of anesthesia in surgical patient. Let us start with describing the dose-response relationship for the first type, the quantitative effect. To understand this relation, we will draw the curve which relates the dose of the drug, on the x-axis, and the magnitude of the effect, on the y-axis. This curve is called the graded dose-response curve. We may also draw the drug concentration, instead of the drug dose, In such case the curve is called graded concentration response curve. Surprisingly, the graded dose or concentration response curve, i.e. the response on the y-axis, and the dose or the concentration on the x-axis, is always hyperbolic in shape, independent of the type of the receptor, or the agonist. If we instead draw the log dose or concentration response curve, i.e. the response on the y-axis, and the log of the dose or the concentration on the x-axis, the curve will turn sigmoid in shape, that is, it takes the shape of the letter S, where the initial and late segments of the curve are nearly flat, while mid-segment of the curve is steep. From the sigmoid curves, we can easily estimate the following parameters. 1. The efficacy, or simply the Emax. This is the maximal effect that can be obtained by the agonist. If you further increase the dose or the concentration, no increase in the response will occur. This may be explained by the fact that, at this point, all the receptors are activated. The efficacy is important to decide which drug you are going to use for each degree of disease severity. For example, if you have a hypertensive patient with mild increase in arterial blood pressure, e.g. the pressure is 10/5 mm mercury above the normal level, then you can use a drug with low efficacy, e.g. a thiazide diuretic. On the other hand, if the pressure is 40/15 mm mercury above the normal level, then you cannot use the thiazides, because their maximal effect is less than the desired response. In such case, you may need an angiotensin-converting enzyme inhibitor, since its Emax is greater than the desired response. Two, the effective dose 50, or simply the ED50, if you draw the dose on the x-axis, or the effective concentration 50, or simply the EC50, if you draw the concentration on the x-axis. This is mathematically equal to the dose, or the concentration, which produces half the maximal effect. It is a measure of the potency of the drug. The potency may be also assessed by the slope of the mid-straight segment of the curve. This slope is mathematically equal to the effect that is produced by the unit dose. The potency is important to decide which dose you are going to use from a particular drug. For example, if an antihypertensive drug has a potency of 5 mm mercury per each milligram of the dose. If you want to decrease the blood pressure by 10 mm mercury, you need 2 mg, and if you want to decrease the blood pressure by 15 mm mercury, you need 3 mg. And so on. We can also draw the relation between the inverse of the response, or 1/E, on the y-axis, against the inverse of dose or the concentration, or 1/D, or 1/C, on the x-axis, to obtain a completely linear curve. In such case, the y-axis intercept will equals 1/E max, and the slope of the curve will equals the ratio between ED50 or EC50 and the Emax. Now, let us describe the dose-response relationship for the second type, the qualitative effect. We sometimes call it the all or none dose-response curve. To understand this relation, we will draw the curve which relates the logarithm of the dose, or the concentration of the drug, on the x-axis, and the percentage of the patients who respond to this dose or concentration, on the y-axis. Again, the curve will take the sigmoid shape. The highest limit of this curve will occur when 100% of the patients respond to the drug. This will, of course, occur at a high dose. Further increase of the dose will not, of course, produce more responders. The dose or the concentration, which produce the desired effect, in 50% of the cases, is called the effective dose 50 or, simply the ED50, if we use the drug dose, and the effective concentration 50, or simply the EC50, if we use the drug concentration. Sometimes, we are interested in studying the drug toxicity, rather than the drug efficacy. In such case, we will do the experiment on the animals, not on human, and we will count the number of the animals, which will die at each dose level, that is, we will draw the relation between the percentage mortality rather than the percentage efficacy, and the log of the dose or the concentration. From these experiments, we will get the lethal dose 50, or simply the LD50 instead of the ED50, if we are using the dose, and the lethal concentration 50, or simply the LC50 instead of the EC50, if we are using the concentration. A new parameter, called the therapeutic index, or simply TI, may be obtained by dividing the LD50 by the ED50. This parameter is a measure for drug safety. The higher the TI, the more safe the drug is. Now, we will postpone discussing the other types of the agonists, the partial and the inverse agonists, till we discuss the antagonists. Let us now discuss the antagonist. If this is the receptor, and this is the antagonist, the three-dimensional shape of the antagonist fits to the receptor, may not be so perfect key and the lock type of fit, as the case with the agonist. However, this is still called affinity. The binding of the antagonist does not trigger any post-receptor signaling. Thus, the antagonist lacks the efficacy. There are two main types of the antagonists. The first one is the competitive antagonist. If this is the receptor, and this is the agonist, the agonist binds well with the receptor. This is the competitive antagonist. It binds to the same binding site as the agonist. Thus, it can displace the agonist from the receptor, to occupy its place. In other words, there is a competition between the agonist and the antagonist, which one will win, and display the other from the binding site. It is the one with the highest concentration. That is, if the concentration of the agonist is higher than the concentration of the antagonist, it will occupy the receptor, and induce a pharmacological action. However, if the concentration of the agonist is lower than the concentration of the antagonist, it will leave the receptor, and its action will be cancelled. One example of the competitive antagonist occurs at the muscarinic receptors between the atropine and the acetylcholine. Another example of the competitive antagonist occurs at the mu receptors between the naloxone and morphine. Now, let us examine the effect of the competitive antagonist on the graded concentration response curve of the agonist. We will use two different concentrations of the competitive antagonist. This red curve represents the concentration response curve of the agonist, with the Emax and EC50, as we have just discussed. This blue curve represents the concentration response curve of the agonist, in presence of a small concentration of the competitive antagonist. The curve has the same Emax, since when we increase the concentration of the agonist, we can displace the competitive antagonist from its binding site on the receptors, and accordingly, we can obtain the same Emax. However, the EC50 is higher, because some of the agonist molecules will be used to displace the antagonist from the binding sites on the receptors. This green curve represents the concentration response curve of the agonist, in presence of a high concentration of the competitive antagonist. The curve still has the same Emax, however, the EC50 becomes higher again, because more of the agonist molecules will be used to displace the antagonist from the binding sites on the receptors. From the above curves, it is apparent that the competitive antagonist displaces the concentration response curve of the agonist to the right. The new curves have the same Emax. However, the EC50 increases. The second one is the non-competitive antagonist. If this is the receptor, and this is the non-competitive antagonist, the non-competitive antagonist binds to the receptor, at an allosteric site, other than the agonist binding site. Sometimes, it binds to the same binding site, but with an irreversible bond, as the covalent bond. If this is the agonist, it cannot displace the antagonist from the receptor, to occupy its place. In other words, there is no competition between the agonist and the antagonist. An example of the non-competitive antagonist occurs at the alpha receptors between the phenoxybenzamine and the norepinephrine. Now, let us examine the effect of the non-competitive antagonist on the graded concentration response curve of the agonist. We will use two different concentrations of the non-competitive antagonist. This red curve represents the concentration response curve of the agonist, with the Emax and EC50, as we have just discussed. This blue curve represents the concentration response curve of the agonist, in presence of a small concentration of the non-competitive antagonist. The curve has a lower Emax, since when we increase the concentration of the agonist, we cannot displace the non-competitive antagonist from its binding site on the receptors, and accordingly, we have less number of receptors to produce the maximal response. However, the EC50 is still the same. This green curve represents the concentration response curve of the agonist, in presence of a high concentration of the non-competitive antagonist. The curve has much lower Emax, however, the EC50 is still the same, because nothing can displace the non-competitive antagonist from the binding sites on the receptors. From the above curves, it is apparent that the non-competitive antagonist displaces the concentration response curve of the agonist downward. The new curves have lower Emax. However, the EC50 is the same. Now, we will return back to our discussion of the remaining two types of the agonist, which we have postponed, till we finish the discussion of the antagonist. We will discuss the second type of the agonist, the partial agonist. Actually, it has features in between the agonist and the competitive antagonist. If this is the empty receptors, and this is the partial agonist, it will bind to the receptor, but the binding is less well fit than the full agonist binding. Thus, the partial agonist still has affinity to the receptors. However, it produces an action weaker than the full agonist. We can say that the partial agonist has partial efficacy. A famous example of the partial agonist is pindolol at the beta receptors and bromocriptine at the D2 receptors. However, what happens when we give the partial agonist, while the receptor is already occupied with the full agonist, which is producing a full effect? The partial agonist will displace the full agonist from the binding site of the receptor. This cancels the action of the full agonist and replaces it with the weak action of the partial agonist. That is, part of the action of the full agonist is antagonized by the partial agonist. Accordingly, we better call the partial agonist the agonist-antagonist, because it acts as an agonist when it is given in absence of the full agonist, and it acts as an antagonist when it is given in presence of the full agonist. The third, and the last type of agonists, is the inverse agonist. If this is the receptor, and this is the inverse agonist, the three-dimensional shape of the inverse agonist fits well in the binding site of the receptor, like the key and the lock. This is called affinity. The binding of the inverse agonist triggers a post-receptor signaling, which initiates the pharmacological response, or the action. However, the triggered action is the opposite to that produced by the full agonist. So, this is called reversed efficacy. The most famous example is the beta-carboline at the benzodiazepine receptors. Another example is the antihistaminic cetirizine, or loratadine, and the H1 receptors. Now, what are the shapes of the graded concentration response curves for the partial agonist and the inverse agonist? If this is the graded concentration response curve for the full agonist, the graded concentration response curve for the partial agonist resembles that of the full agonist in the presence of a non-competitive antagonist. And the graded concentration response curve for the antagonist is a horizontal line. And the graded concentration response curve for the inverse agonist is the mirror shape reflection of the partial or full agonist on the concentration axis. Concentration axis. This presentation about the pharmacodynamics is prepared by Professor Mohammed Abdelbari, Professor of Pharmacology, and Shamas Faculty of Medicine. The prepared narration is read by the Microsoft David voice. The term pharmacodynamics may be defined as the effect or the action of the drug on the body. We are interested in two main items when we discuss the pharmacodynamics of a drug. 1. The mechanism, or the mechanisms, by which the drug induces its actions. 2. What are the factors which may modify the drug actions? This will be postponed to be discussed with the pharmacotherapeutics. By the end of this lecture, you should be able to 1. List the different theories that explain the receptor mechanisms of drug action, and mention the significance of each one. 2. Enumerate the details of signaling mechanisms of drug action, with examples. 3. Enumerate the non-receptors targets for drugs, with examples. 4. Enumerate the physical and chemical mechanisms for drug action, with examples. Now, let us try to explain the mathematical relation between drug binding and the response. The first theory was that of Clark, the receptor occupation theory. It postulates that the response to a ligand is directly proportional to the number of the receptors occupied by the ligand. If this is the total number of the receptors on a cell, if 25% of the receptors are occupied by the ligand, then we will obtain 25% of the maximal response. If 50% of the receptors are occupied by the ligand, then we will obtain 50% of the maximal response. The maximal effect, Emax, will occur when all the receptors are occupied. However, there are too many weak points in this theory. One of them is that, sometimes the maximal response occurs after binding of only a small number of the total receptors, e.g. full hypoglycemic effect of insulin, occurs after binding of only 5% of the total number of the insulin receptors. This leads to the postulation that there are spare receptors, which do not share in the action. These spare receptors are important, since they act as a reserve. These spare receptors may explain the delayed onset of type 2 diabetes mellitus. The occupation theory still has weak points. One weak point is that, sometimes, the maximal response to a certain agonist did not occur despite binding of the total number of the receptor on the cell by this agonist. A modification was made by Peyton. It is the rate of binding of the agonist to the receptor, and not the number of the receptors that bind the agonist, that determines the effect. Thus, one molecule of an agonist can activate the receptor more than once. Each time it associates with the receptor, it will activate it, then it dissociates away, to reassociate with it again and to reactivate it. If this is the total number of the receptors on a cell, repeated association and dissociation of the agonist can activate the same receptors many times. After the rate modification of the occupation theory, we can postulate a good mathematical relation for the effect of the drug concentration on the drug response. The effect of the drug, E, equals the maximal effect, or the Emax, multiplied by the drug concentration, and divided by the sum of the drug concentration plus the EC50. We can also explain the tolerance that occurs to the agonist by the phenomenon of downregulation of the receptor's number, and that occur to the antagonist by the phenomenon of upregulation of the receptor's number. However, there are still points that need to be explained, e.g., the effect of the partial and inverse agonists. Accordingly, the two-states model theory was introduced, and it was followed shortly by the three-states model theory. The two-states model theory postulates that the receptor may be present in one of two states: the active state, Ra, which can induce a pharmacological action when it binds to a ligand, and the inactive state, Ri, which does not induce any action when it binds to a ligand. The ratio of the two states is responsible for the net effect of the ligand. The antagonist binding shifts the equilibrium to the inactive state, so it produces no effect. While the agonist binding shifts the equilibrium to the active state, so it produces an effect. The three states model theory was soon introduced to complete the picture. The three states model theory adds a third state, which is the constitutive state, or R-hash. This state has some activity, even in absence of ligand binding. Similar to the two states model theory, the antagonist binding shifts the equilibrium to the inactive state, and the agonist binding shifts the equilibrium to the active state. The inverse agonist binding shifts the equilibrium away from the constitutive state, thus it produces an effect opposite to that of the agonist. Now, the second component of the receptor action, the signaling. We have 5 types of signaling mechanism. Let us start with the first type, the receptors gating the ion channels. There are 2 famous examples, the GABA receptors, which gate the chloride channel, and the nicotinic NM receptors, at the motor end plate of the skeletal muscles, which gate a mixed sodium-potassium channel. This is a sagittal section of the ion channel, which is made of 5 subunits, alpha 1, alpha 2, beta, gamma, and delta subunits, with the ion channel surrounded by the innermost part of the 5 subunits, and is closed at rest. The section shows only 2 subunits. This is the cell membrane of the skeletal muscle, just opposite to the cholinergic motor nerve. The ion channel is transmembranous, that is, it is crossing the cell membrane, with the outer part in the extracellular fluid, and inner part in the intracellular fluid. This part of the cell membrane is called the motor end plate, or simply MEP. The extracellular fluid contains excess of the sodium ions, while the intracellular fluid contains excess of the potassium ions. This blue triangle represents the acetylcholine, released by the motor nerve. It binds to a site between the alpha subunit and the adjacent subunit on the nicotinic ion channel. Binding triggers opening of the ion channel. This allows the extracellular sodium ions to enter the cell, and the intracellular potassium ions to escape from the cell, at the same time. The net effect of movement of the positive ions is a slight depolarization of about 5 to 15 millivolts at the motor end plate. This depolarization is called end plate potential. It can triggers action potential in the nearby muscle membrane. The action potential, in turn, initiates skeletal muscle contraction. The acetylcholine is rapidly hydrolyzed by true choline esterase enzyme, which is present at the nearby muscle membrane. This terminates the action of acetylcholine, and the ion channel recloses. Repolarization follows, by opening of a voltage-operated potassium channel, as well as by activation of a sodium-potassium ADPase. Careful observation of the above steps shows that this mechanism is a very fast mechanism, as it depends on rapid potential changes. The second type, the receptors linked to tyrosine kinase enzyme. The most famous example is the insulin receptors. This is the cell membrane. And this is the insulin receptor homodimer, That is, the insulin receptor is made of two identical parts. Each part is made of two subunits, a transmembrane alpha subunit, which contains the insulin binding site, and an intracellular beta subunit, which possesses the tyrosine kinase activity. This is the insulin hormone. Binding of the insulin triggers aggregation of two parts to form the dimer. This dimerization activates the tyrosine kinase enzyme, leading to autophosphorylation of the tyrosine residues on the beta subunits. The activated phosphorylated dimer binds to relay proteins, activating them, which in turn by cascade effect, can activate up to 10 others. These relay proteins are called insulin receptor substrates, e.g., IRS1 and IRS2. These insulin receptor substrates mediate the actions of insulin through phosphorylation of other cellular components, e.g., glycogen synthase, which initiates glycogen deposition in the liver, or hormone-sensitive lipase, which inhibits fat cell lipolysis, etc. A subtype of the tyrosine kinase receptors is the JAK receptors. It differs from the parent family in that the receptors do not possess a tyrosine kinase activity, but is linked to a special type of tyrosine kinase, called Janus kinase, which is abbreviated JAK. Similar to the parent tyrosine kinase receptors, they undergo dimerization on binding of the ligand, e.g. the cytokine interferon. However, no autophosphorylation occurs, but just activation of the JAK. JAK, in turn, phosphorylates tyrosine residues of several signal transducer and activator of transcription, STAT proteins. Two of the activated STATs undergo dimerization. And then, the dimer is translocated to the nucleus to control transcription. The third type of the signaling mechanisms is the non-membrane bound receptors, which regulate the transcription. The most famous example is the steroid receptors, e.g. the receptors for cortisol, aldosterone, vitamin D, etc. The ligand which activate these receptors should be highly lipophilic to cross the cell membrane, since these receptors are located in the cytoplasm. This is the cell membrane. This is the nucleus. These are the genes, carried on the double-stranded DNA molecule, inside the nucleus. These are the cytoplasmic steroid receptors. In the inactive state, the heat shock protein, HSP, occupies the ligand binding site. These are the cortisol molecules. They penetrate the cell membrane to bind to their receptors at the ligand binding domain, after displacing the heat shock proteins. This is followed by dimerization to form the active homodimer, which is translocated to the nucleus to bind to the DNA by its DNA binding domain. They activate the DNA by their nearby DNA activating domain, with subsequent formation of the messenger RNA. These are the ribosomes, present on the rough endoplasmic reticulum. The messenger RNA is translocated to bind to the ribosomal RNA. This will initiate a new protein synthesis. It is called lipomodulin, or lipocortin. This protein mediates the action of cortisol. Of course, this mechanism of signaling is the slowest signaling mechanism, so drugs acting through transcription regulation are never life-saving in emergency. The fourth group of signaling mechanisms includes a family of proteins, called the G proteins. This is the cell membrane. This is the enzyme, adenylyl cyclase. This is the G protein receptor, the beta-adrenoceptor. It is a polypeptide made of 7 transmembrane domains, with 6 loops connecting these transmembrane segments, 3 extracellular loop and 3 intracellular loop. The C-terminal end of the polypeptide is the cytoplasmic tail, while the N-terminal end of the polypeptide is the extracellular tail. This is the epinephrine, the ligand of the beta receptors. It binds to a site surrounded by the 7 transmembrane domains. This is the GS protein. In the inactive form, it is bound to GDP. Upon binding of epinephrine, the G protein moves to bind to the 3rd cytoplasmic loop of the beta receptor. This binding converts the GDP into GTP. The GS protein is said to be activated. The alpha subunit of the active GS protein is released to bind to the adenylyl cyclase to activate it. The active adenylyl cyclase enzyme converts the ATP into cyclic AMP. The cyclic AMP acts as a second messenger. The most important function of cyclic AMP is phosphorylation. The cyclic AMP induces phosphorylation of the regulatory subunit of the enzyme protein kinase A. The control subunits of the protein kinase A enzyme get released to initiate a cascade of phosphorylation of cellular targets. The GI protein is similar to the GS in all respects, but it inhibits the enzyme adenylyl cyclase, thus it has the opposite actions to the GS protein. It is linked to the adrenergic alpha-2 and the muscarinic M2 receptors. Another type of G proteins is the GQ protein. It differs from the GS protein in the following respects. It is linked to the enzyme phospholipase C instead of adenylyl cyclase. The most important examples include the alpha-1 adrenoceptors and the muscarinic M1 and M3 receptors, not the beta adrenoceptors. Binding of the active alpha subunit of the GQ to the phospholipase C enzyme leads to activation of phosphorinositol diphosphate into inositol triphosphate, or simply IP3, and diacylglycerol, or simply DAG. Both act as second messengers. IP3 stimulates the sarcoplasmic reticulum. leading to calcium release to the cytoplasm. This calcium activates many cellular processes. This enzyme is called protein kinase C. It is stimulated by both the calcium ions, as well as the DAG. This leads to a cascade of phosphorylation. The last type of signaling is that used by the nitric oxide, or simply NO. The nitric oxide has its own receptors, similar to that of the neurotransmitters and the hormones. The receptor is present intracellularly, and the nitric oxide diffuses through the cell membrane, to bind to a metal ion, or to one of the sulfur atoms, of the amino acid cysteine of the receptor. Before discussing the signaling mechanism, we need to know the source of the nitric oxide. It is synthesized from arginine, by the enzyme nitric oxide synthase, or simply NOS. The nitric oxide is synthesized in many cells in the body, mainly the endothelial cells, where it was called the endothelial-derived relaxing factor. The endothelial nitric oxide synthase is activated by the M3 muscarinic receptors, as well as by the bradykinin receptors. The nitric oxide may also come from an external donor, e.g. the nitrovasodilators, nitrates, and sodium nitroprusside. The signaling mechanism of the nitric oxide receptors is similar to that of the autonomic neurotransmitters, that is the G-protein coupled receptors. However, the linked enzyme is the guanyl cyclase enzyme. It converts GTP into the second messenger, cyclic GMP. The function of the cyclic GMP is similar to that of the cyclic AMP, that is, phosphorylation. However, the target here is the protein kinase G. The activated protein kinase G induces calcium efflux out of the cell. The decrease in the calcium concentration is responsible for the actions of the nitric oxide. Interestingly, we can simulate the actions of the nitric oxide by increasing the cyclic GMP levels in the cells. The most famous example is the sildenafil, which increases the cyclic GMP by inhibiting its degradation with the phosphodiesterase enzyme. Now, the drugs that act on non-receptor targets. Examples of drugs acting through enzymes include aspirin, which inhibits the cyclooxygenase enzyme, or simply, the COX enzyme, and omeprazole, which inhibits the potassium hydrogen AT-base enzyme, or simply, the proton pump. Examples of drugs acting through the cell wall include the antibiotic, penicillin, which binds to penicillin-binding proteins, to inhibit peptidoglycan synthesis, which is the main component of the bacterial cell wall. Examples of drugs acting through the cell membrane include the pulling antifungal drugs, e.g., nystatin, which binds to ergosterol of the cell membrane, to create pores. Cellular constituents are lost from these pores. Examples of drugs acting through the microtubules include the colchicine, an anti-inflammatory drug used in gout, binds to the tubulin protein of the neutrophils to inhibit their migration to inflammatory sites. Examples of drugs acting through the ribosomes, include the tetracyclines, and the aminoglycosides antibiotics, which bind to 30S subunit of the ribosomal RNA. Examples of drugs acting through the mitochondria include the anti-helminthic drug, niclosamide, which uncouples oxidation from phosphorylation. The last mechanism for drug action include the chemical and physical actions. As regards the chemical mechanism, we have many examples, e.g., the antacids, which neutralize the gastric hydrochloric acid to promote healing of peptic ulcer. The protamine sulfate, which is used for treatment of heparin overdose, it neutralizes the negatively charged heparin by its positive charges. The chelation, which means the capacity of organic compounds to form complexes with metals to form chelates. These chelates are more water-soluble, and thus are easily excreted. This is useful in treatment of heavy metal poisoning, e.g., British anti-Lewisite, or bale, in mercury poisoning, and penicillamine in copper poisoning. As regards the physical mechanisms, we have the demulcents, and the emollients, e.g., olive oil, which is used as demulcent for dry mucous membranes, and as emollient for dry skin. The adsorbents, e.g., choline, and pectin, which are used in diarrhea. They are adsorbed to the inflamed intestinal mucosa, to arrest the discharge. The lubricants, e.g., liquid paraffin, which is used in constipations, to facilitate expulsion of the hard stools. The drugs which act by their osmotic effect, e.g., the osmotic purgative, lactulose, and the osmotic diuretics, mannitol. Mannitol.